All right, thank you everybody for coming back after lunch. So we have a 30-minute talk now, and then we will move over to the Australasian Mycological Society talk. So the next speaker is Laura Van Galen, who some of you might remember from the 2018 Fungal Foray when she first arrived in New Zealand from the University of Tasmania, and she's just handed in her incredible PhD. So she's going to tell us about some of the things that she did for that work. Over to you.
Thanks, David. Yes, today I'm going to talk about part of my PhD, and my PhD was about the relationship that forms between Nothofagus species and the ectomycorrhizal fungi. And it was about understanding how the fungi affect the tree establishment and therefore forest restoration practices, but also how the trees and the environment affect the fungal communities. So I'm going to talk mostly about the fungal component of my work. Firstly, the Nothofagus species, quite a few people have talked about these already. The southern beech, they're all throughout New Zealand. In fact, 70% of native New Zealand forests have beech species in them, but they're also in South America, as Camille was talking about, in Tasmania and Eastern Australia and New Guinea and New Caledonia. And they're split up at the moment into 4 subgenera. In New Zealand, we've got 5 Nothofagus species. Well, 2 of them are subspecies, but we've got the Salandrii species, Cliffordioides and Salandrii, the black beech and mountain beech. We've got Nothofagus menziesii, silver beech, Fusca and truncata, red beech and hard beech. And as for the subspecies, we've got Nothofagus menziesii in the Lophozonia subgenera, and the other 4 are in the Fuscospora subgenus. And so the Lophozonia subgenus is much older than the Fuscospora one. Just remember that for later on. But all Nothofagus species are obligately ectomycorrhizal, so they can't live without the ectomycorrhizal connections. And as we've heard a bit about today, there are lots of different types of mycorrhizal connections, and they differ in how they associate with the plant roots and what they can give the plants. Arbuscular mycorrhizae are very widespread, and there's huge numbers of host species that associate with them. And the best guess that I found is there's about 300 to 1600 fungal species that form arbuscular connections. But on the other hand, the ectomycorrhizae, there's a lower diversity of plant. So in New Zealand, it's only the Nothofagus and the Leptospermum, which is the Manuka, and the Kunzea, the Carnica, that form ectomycorrhizal connections, and then also the introduced pines and eucalypts. But a lot of these trees dominate forests across the world, so they're still hugely ecologically important. And interestingly, the estimates of ectomycorrhizal fungal species diversity is hugely higher than the arbuscular, 20 to 25,000 fungal species. So it's a pretty interesting group. In New Zealand, luckily, we know quite a bit about the ectomycorrhizal fungi from the fungal forays, which have been happening for a very long time, where people have gone out, as everyone here knows, and collect a lot of fruiting bodies from around the country. And this has given us a great understanding of all of these different fruiting species and given us specimens for sequencing. So we've got a good reference database. But obviously, the fruiting bodies is just the tip of the iceberg in terms of the actual fungi in the soil associating with the trees. And because they only come up every now and again, and you've got to happen across them at the right time, they leave a lot of holes. There's a lot that we don't know about the fungi in New Zealand. We really don't know how many fungal species there are. As Sam was talking about in some of his estimates, there's a lot more that have been undetected or undescribed. We don't know what they are. We don't really know how they're affected by the environment. So do they respond to soil properties and rainfall and temperature and other forest structure factors? And we don't really know how they are between the different types of host species. So that's something that for my PhD, I wanted to dive into a bit more. And so to do this, I looked at environmental DNA. So what's going on actually underground in the soil. And I used hyphal ingrowth bags to do that. These are just small mesh bags filled with sand, and they're really fine mesh so that tree roots can't grow inside, but fungal hyphae can grow in. So what you do is you bury them in the ground. You can see the pink tape poking up here where it's buried. Leave them in there for multiple months. Fungi will grow through the bags, and then you can come and collect them again, and you've got a nice little package of fungal hyphae that then you can sequence the DNA of, and it's a nice sort of package of what species are there in the soil. So I buried these bags around the country. So this is the data that Sam was talking about that he was using before. So I buried them at 81 different sites, and these sites were dominated by different Nothofagus host species. Some of them were dominated by more than one, the mixed sites. And in each site, I set up 10 different plots. And then in each plot, I buried 5 different hyphal ingrowth bags. So that works out to be 50 bags per site, which was 4,050 in total. But then I pulled the samples from each of the plots for the DNA analysis. So I ended up with 110 final samples. So the forests that we were visiting ranged hugely in their environmental characteristics. There were these really dry, classic, open, monodominant beech forests through to these very wet, full of ferns and other vascular plants. But all my sites were dominated by Nothofagus, and they didn't have any other ectomycorrhizal host species present. Whilst we were there, we collected soil samples to do soil chemical and physical property analysis. I looked at the ground covers, the bryophytes and ceilings and other things, collected data on the canopy cover, the light getting through, the age cohorts of the trees, the forest age data, climate data, rainfall and temperature, and then also I calculated the measures of patch size to understand how the sort of forest fragmentation might affect communities. The bags were in the ground for 9 months, after which we went back to try and find them again. It was quite a challenge. As you can see here, there's a bit of pink flaking tape poking out from under the bryophytes, but it's been a bit overgrown. But actually, all in all, it worked really well. 97% of the bags were able to be recovered. Some of them, like this one in this picture up in the top right, had been pulled out of the ground by animals or other things. But actually, of the ones we were able to recover, 92% were still in the ground. So this was pretty successful, really. Some areas were very muddy. This is Kate here trying to dig the bags out of the ground from the mud. But yeah, it was quite variable. And then back in the lab, we emptied out the sand and added a bit of sterilised water and shook them up. And then the hyphae and the sand floated apart. The hyphae floated on top of the sand, and I was able to extract that out and macerate it all up with sterilised scissors and then take a subsample for DNA extraction. And so then I sequenced the ITS2 region and sent them to Massey University for Illumina MySeq sequencing. And so then all the raw sequences were returned to me of everything that was detected in those bags, and I used the Data2 pipeline to produce these ASVs. So Sam mentioned those, the amplicon sequence variants. These are a measure of all of each one is a separate sort of sequence variant that was present in the sample. So it's kind of similar concept of species, but this is a much finer resolution. So multiple ASVs can belong to the same species. And I use the Unite and the fungal databases to name all of the ones. Obviously, things we don't have in the databases, we can't name, so they just have to be chucked out, but that's the best we've got. So in total, there was 39,500 ASVs, and 5,229 could be classified as ectomycorrhizal using those databases. So that's only a small percentage of all of the ones that were identified, but those ectomycorrhizal ASVs actually account for 60% of the sequence reads. So that's an indication that those hyphal ingrowth bags were selecting for the active ectomycorrhizal fungi in the soil. So this round diagram here is a corona chart showing the sort of the taxonomic breakdown of the samples. And so there's the phyla in the middle all the way through to the genera around the outside. And so we had a huge number of the ASVs were identified as Gordonnaria species, which makes sense based on, we know they're very prevalent in New Zealand. But interestingly, there was quite a lot of diversity associated with this Roulandiella genus, which hasn't actually been detected in New Zealand, I don't think, before. It's in South America and other places. But that's from the Ascomycota. But there was also a lot of Anospes, Lacarius, Tomentellas, things that we know are around. So I wanted to firstly look more at the environmental effects controlling the community assembly of the ectomycorrhizal fungi. So to do that, I used this relatively new technique called multi-site generalized dissimilarity modeling. And this allows you to compare the relative importance of different environmental variables for driving the turnover in fungal communities. So community turnover is just how much change you get as you go from one community to the other, how many shared and different species there are. So I'll explain what all these lines are in a minute, but the y-axis here is the amount of community turnover that's occurred. So high values, variables with high values mean that they're causing a lot of change. And the x-axis is the explanatory variables rescaled. So you can see at what point along the environmental gradient the change is happening. So this dark blue line here is annual rainfall. And you can see that a lot of change in communities is happening as you go from very low rainfall to sort of medium low rainfall, but then less change in the community happens as you go from medium to higher rainfall. And so I was comparing sort of the environmental variables, which are the solid lines, to the some host variables, which is the host species that were present at the site, the dashed lines, and then the red dashed line is the distance between the sites. So to get an understanding of how the geographical distance between sites might affect species. So things that are sites close together might share more species because it's easier to disperse. So firstly, the soil carbon to nitrogen ratio and soil pH were having strong effects on the changing communities, so the community assembly, as well as the annual precipitation and the forest patch size. These were the strongest drivers. As for the host variables and the distance variables, these were also having medium effects on the species distributions. The two host variables that were important was the percentage cover of Nothofagus menziesii, which is the silver beech, and also the Nothofagus salandri varclipitioides, the mountain beech. So one thing that's kind of hard to get your head around, looking at turnover between pairs of sites, is actually heavily influenced by the rare species in the community. But what this multi-site generalised dissimilarity modelling allows is for us to look more deeply at the other parts of the community, so the more common species. And so instead of just comparing pairs of sites, we can take groups of large numbers of sites, so groups of 3 sites or 4 sites or however many, and look at what species are shared between those sites. And that gives us an understanding of what's driving the common species turnover. So I looked at groups of sites all the way through to group of 8 sites. That's what order 8 means. And there are actually some the variables that are important change as we look at increasingly more common species. Firstly, the carbon to nitrogen ratio and pH is still quite important. pH has dropped a little bit, but it's still up there. The precipitation and forest patch size is having a less of an effect on the common species compared to the rare species. Instead, it's the mean size of the Nothofagus trees and the annual temperature range that's driving this common species turnover. Also isothermality, which is kind of hard to explain, but that's another temperature variable, and the slope of the site are also having an important effect on the common species turnover, whereas they weren't doing much over here at the rare species. And our host variables and distance between sites are now having very little effect on common species, which is unsurprising because we might expect more common species to be more generalists and distributed everywhere. So kind of the take-home message from here is that there's this interesting mix of soil variables, climate variables, and then host tree characteristics that are controlling the communities, but that changes depending on if you're looking at the rare versus the common species. And so I don't think this method has been applied to fungal communities before, but this is really important to be thinking about this because we need to know how these communities will change as the climate changes in the future. Now look, the variance explained by the different types of variables in more detail. And you can see that the blue, the environmental variables, were explaining most of the variation in the model for both the rare and common species. But the host variables were still explaining a little bit of the rare species. And so this 3.8% here indicates that this variation can only be explained by the host species at the site. So even though environment's having a strong effect, host is still having some effect. And so that's kind of what I looked at next. So I mentioned before that we have 5 species in New Zealand. And so firstly, I looked at the richness, just the number of ASVs present, but there wasn't really a pattern there. And I also looked at the mean phylogenetic diversity, the MPD. So that's not just the number of species present, but how much genetic diversity is present. And interestingly, the Nothofagus cliffordioides, the mountain beech, had lower phylogenetic diversity than the rest. And so that's kind of an indication that they're more specialized or more closely related, the fungal species in the mountain beech compared to the rest. When looking at the subgenus level, so the Nothofagus menziesii is the Lophozonia subgenus and the rest are in the Fuscospora, there was actually a stronger pattern. There's this trend of greater richness and phylogenetic diversity in the older Lophozonia subgenus. But what became marginally non-significant when I included environmental covariates, because these host species also have their preferred environmental ranges. And I'll talk a bit more about that in a minute. But there's still a decent trend there. And we also looked to see whether having more than one host species present at a site might increase the diversity, but there wasn't strong patterns there either. So I then looked at the overall community composition and not just the number of ASVs. And here is where some really interesting patterns arise. So these are ordinations. The left is the taxonomic ordination and the right is looking at the phylogenetics. And so each of these dots is one of the sites, and dots close together have similar communities and dots further apart have more different communities. And they're coloured according to the Nothofagus species, the dominant Nothofagus species at the site. And so you can see straight away that the colours are clustering together. So the communities from sites dominated by the same host species are more similar than those dominated by different host species. And so when I clustered those communities with these dendrograms in the corners, and really interestingly, there was this strong pattern of co-phylogeny where more closely related host species had more closely related fungal communities. So up in the top corner here is sort of a representation of the relationships between the Nothofagus species. And so as I mentioned, the Lophozonia is much older than the rest. And particularly this phylogenetic ordination here, sadly I couldn't look at Nothofagus truncata because there wasn't enough replication, but the other ones, the fungal communities are clustering in the same pattern as the relationship between the hosts. And when I did a statistical test for that, it was highly significant. So this is pretty cool because this often happens in like host parasite communities, and it can be an indication of coevolution, but a lot of other things can cause this matching up of phylogenies too. For example, if more closely related host species have more similar phenotypes, that they then select for more closely related fungal species. But it's really cool that this pattern has emerged amongst these Nothofagus species, given that the Nothofagus species are all quite closely related too. And lastly, again, I looked at this in a bit more detail, and I did some randomization tests to determine whether or not the fungal communities of the four main host species were phylogenetically specialized. So this top figure here is the phylogenetic diversity, and anything below the line means that they're more specialized or like less phylogenetically diverse than you would expect by chance. And all four groups were, so that's more indication of specialization. But then I also looked at this second one, the relative phylogenetic diversity, and this just compares the branches on your tree to a tree with equal branches. And so it can give you an indication of if the branches are longer or shorter than expected by chance. And so for the three Fuscospora species, there was no significant difference, but for the Lophozonia species, sorry, the Lophozonia communities, they had longer phylogenetic branches than you might expect by chance. And so interesting that that matches up with the Lophozonia being the older host lineage as well, which is exciting. So before I mention environmental covariates, because some of the host species have specialized to different environmental conditions, and there was actually differences in the elevation between the sites dominated by different host species and in the seedling cover. So I wanted to look at this in more detail because it's not possible to include covariates in the analyses looking at the like the ordination and the phylogenetic patterns. So instead I did a separate analysis using a generalized dissimilarity model to look at how these variables explain the variation in phylogenetic turnover. So this is the same concept as what I did with the environmental variables earlier, but this is simplified. It's sort of the original version of the method, and it's looking at the phylogenetic turnover, not the like the taxonomic turnover. And so what that showed was that the identity of the host species at the site explained much more variation than the elevation or the seedling cover. So even though there was still some variation explained by elevation and seedling cover, and we're not sure what of that or what of the differences in the fungal community could be related to that, the fact that the host identity is explaining so much more gives a bit more added confidence that these patterns I'm seeing are due to host effects and not just sort of shared preferences between the fungi and the host for different environmental conditions. So in summary, there are different environmentals driving the community patterns of rare and common ectomycorrhizal species. Soil variables seem to be quite important for everything, but the forest patch size, precipitation, and the host species have a greater effect on the rare fungal species, whereas the temperature variables and the host tree size were affecting more common species. And it seems that in New Zealand, the different host species are harbouring different fungal communities, and there's that really interesting pattern of cophylogeny and of the branch lengths. So what's next is, it will be really cool, I think, to dive into this host specificity question a bit further and try and do some more experiments to show that these phylogenetic patterns we're seeing are actually directly due to host effects. Data set also provides great potential for a lot of biogeographical analyses, and I'm hoping to try and look at patterns of diversity and endemism and how they change across the South Island. And hopefully this eDNA data can be used to map fungal species distributions, like what Sam was talking about, and get a sort of use this as a new method to supplement the fruiting body collections and try and get a better understanding of the species that we've got here and which ones might be under threat. A huge number of people were involved in this project and helped with all the field work and the lab work. And so thanks very much to all of them. Thank you.
Great. Thanks, Laura. Gosh, that's good too. Well, I already knew that, so I shouldn't sound surprised. Great. So thank you. Hopefully that will answer your question, Peter Johnston, about host specificity. I think we should open it up for questions. Hopefully. Oh, Camille, right, good one. I'm just going to let you, right, you should be able to turn your stuff on now. Yep.
Hi, Lara, very great talk. Thank you so much.
Thank you.
I think I was interested by the pattern that you found with this the Rullandiella ascomycetes that are ectomycorrhizal. And I was wondering whether you or anyone in the group during the foray, if you guys have found spore mats of this genus. So I don't know if you're familiar with this. The fruit body of Rullandiella, it's this like really small upside down truffle. It looks like an insect egg. It's really hard to find it in the soil. But in Patagonia, we found that this group, they heavily produce mitotic spore mats. So it's like for asexual reproduction. It looks like a small puffy little mat on the soil. And this is actually how we realized that especially one of the species, Rullandiella patagonica, was really abundant in the soil, not based on the food bodies, but based on the spore mat. So I was wondering if this is a pattern that you have observed in New Zealand as well, because in Tasmania, I haven't not seen that many actually. So yeah.
Yeah, I haven't seen what you're describing. I don't know whether you have, David.
No, I don't think I have any clue what to look for, and had never heard of it until Laura got it. Yeah. So, and I mean, from memory, there's quite a bit of phylogenetic diversity in those sequences you have, Laura, isn't that right? And
yeah, so that's why
it maybe
that's
not match so much with,
showing.
yeah, and did it match much with taxa that are already known elsewhere?
That's something that I need to look at further. Yeah, doing some more phylogenetic work on how my sequences compare to the current type specimens. And I did some preliminary analysis, and it was a bit inconclusive. Yes, so I don't want to say too much because I don't think I did a very good job of it. So need to do a better job of it.
Yeah, I had a vague inkling that maybe that name may not even be applicable. Like there could be something related to it, or that.
Yeah.
No, there is a taxonomic paper that was done recently where they described actually the species based on the fruit bodies. And this group is found abundantly with seedlings, actually, like for example in nurseries. So it is possible that in GeneBank, a lot of the sequences are actually identified as like PETZAC. Not all of the environmental sequences are identified, but this is a well-defined genus. I can see if I can find some publication if you want, but yeah, this thing with the sporma is, I think it's really interesting because we really found the spormas everywhere in Patagonia.
Maybe get some photos because then you could send them.
Yeah,
Could have a look out for them.
There is a publication like about the general, spormats. I can put it in the chat if you want. So I think that would be, yeah, it'd be interesting to also look for them, you know, because this is a really good way because you can sequence the DNA from the spormats. So this is a really good way to find like the distribution of these species not only based on like the soil metabarcoding. Thank you so much. That was really great talk.
Right. Okay. Thank you. That's awesome. So David, you can have a go now.
Yeah, a question, and you can choose to say that you're not going to answer because it's not your area, but I was wondering if there's any indication whether the data you produced for the beach is reflected or may be reflected in the Manuka Kanuka environments in New Zealand.
Yeah, that would be super interesting. And that would be amazing if I could get fungal eDNA data for the Manuka and Kanuka, which I just haven't got. But that's kind of like next on the list of things to get, which would be really cool. Awesome.
Good to hear it.
Yep. We have a plan that Megan Burton, who's doing her master's, is going to do some high-filling growth bags there. So, yeah, that might be something too. Okay, good. Right, Susie said, great information. I need to digest some of this. Yep. Peter J said there's a little bit of unpublished data out there. I guess you're meaning with Rolandiella. Oh, on Manuka, right? Yeah, okay. So Manuka sequencing. Yep. Good. Right. Okay, fantastic. Anything else? No. All right. I'm just going to say thank you again to Laura for, as usual, a great talk.